So this is Gigaohm News. Gigaohm News is high resistance, low noise news brought to you by a biologist. Tonight we are going to discuss the unusual features of the SARS-CoV-2 genome, suggesting sophisticated laboratory modification rather than natural evolution, and delineation of its probable synthetic route. The last line there is so awesome. The authors of this paper tell you that they are going to delineate, they are going to explain to you how it could have been made synthetically, step by step. So this paper is a pretty big swing for the fence. I'm very happy that they've done it, and I'm also very happy to put a name to the famous anonymous scientist and author of the Nerd Has Power blog, Shu Kang. I apologize if I'm saying your name wrong. But the Nerd Has Power blog was very influential in making me early on become convinced that we're having a party with something that came from a laboratory, and that blog was cited by many folks and continues to be cited by many folks. And so this is the lady. This is the lady that was on Fox News, and if you hear her talk, she kind of has this very nasal and loud voice. It's almost like she thinks she has to yell into the microphone all the time, but this is the first time we're seeing anything. I notice here right now that she's an MD, PhD, medical doctor, and a virologist, so that's pretty great. At least on paper, she seems to be a very accredited woman, and so it's been hard to take her seriously only because she's only been on Fox News, right? We're not seeing her on PBS NewsHour. Nobody is really interviewing her. So your subconscious doesn't want to really take her seriously because she's only talking to Steve Bannon on that show that he does, or doing stuff on Fox News. But actually, she's worth taking seriously. And again, Shu Kang is the Nerd Has Power blog. If you remember the Nerd Has Power blog, this is the blog where he showed how many non-synonymous mutations were present in SARS-CoV-2 compared to these two viruses that are cited in this paper. And so that blog was really a big step forward early on in this discussion. And so I'm really happy that that's there, and that they're working together to do this. I don't know who these other two authors are, but good for them, and I'm really happy. And so let's get into this. You're not going to believe it. So I'm just going to, I put a lot of the text up here because, we're reading it together, but I'm going to try to explain what I've gathered from it, and hopefully I won't waste too much of your time by reading this. But they're going to present 3 lines of evidence to support their contention that laboratory manipulation is part of the history of SARS-CoV-2. So very clearly they're already saying that this is not a virus that was created from scratch. And so, you're right, Michael Osterholm, it wasn't a gene jockey didn't come up with this sequence, but it's a genomic sequence of SARS-CoV-2 is suspiciously similar to that of a bat coronavirus discovered by the military laboratories. And now these military laboratories, of course, found it most likely in the cave that we've talked about before in Moyang province, Moyang County in Yunnan province of China. This is where the 6 miners broke out with what was COVID-12, I guess, but basically they broke out with a SARS-CoV-2, a COVID-19-like illness in 2012, and the Army went there and sampled from these miners, including the 3 that survived and the 3 that died. And so it's not really sure which one of these viruses is which one of these things. It could be that the virus they're talking about here in this paper is that virus. It could also be just a different one from the same sort of sampling time period that we've been interested in for these last few weeks. The receptor binding motif, this is the part of the spike protein that actually binds and interacts with our own proteins, which determines the host specificity of the virus, resembles that of SARS-CoV from the 2003 epidemic in a suspicious manner. And this is something that a lot of people have pointed out offhandedly But because most of these people like Christian Anderson and Peter Daszak have pointed to this as evidence for the common sort of ancestry of these viruses, it's been very difficult to turn it on its head. But this paper does a very good job of doing that, which is another plus in its hat. SARS-CoV-2 contains a unique furin cleavage site. We've talked about this before, but I haven't talked about it too much with you guys. But lots of people have been identifying this as one of the things that we need to be aware of as indicating as indicating that it's it doesn't fit in with the natural history of coronaviruses that we'll see as we move forward. And I think that's what they're saying here. I'm not going to read that over to you. So 1.1, the first series, this ZZ45 here is this virus right here is the virus that one of the two viruses that was discussed in the Nerd Has Power blog. So the Nerd Has Power blog compared this one to another one. It might have even been this ZXC21, but I don't think so. I think it was more like 45 and 47 or something like that. But it's irrelevant. It could have been this one. The point is that ZC45 was one of the viruses that was identified, the sequences that was identified by the Nerd Has Power blog as having a ridiculously high homology to SARS-CoV-2, and specifically in a way that didn't make sense, where the changes that were the differences that were found in functional regions were actually differences that didn't make any difference. They were non-synonymous. There were very little non-synonymous mutations, you see. So that's a anyway, we can cover that again some other time, but the structure of this 30,000 nucleotide long genome is shown in figure 1, and searching that database reveals, among all other at least 2 related coronaviruses that share the highest sequence identity with these. But RATG13 was the one that was, of course, reported. And so the note that the RATG13 virus is excluded from this analysis, and so this analysis is in the next figure here. Hold on a second. I'm going to check this live chat and see what they're talking about. There had an autopsy of these miners. There was no proper investigation. No, that's true. That's what we said in previous videos. Anyway, so here's this thing, and what you have to be able to see is right here, where ZZ45 and SARS-CoV, the two SARS-CoVs here, all branch off and aren't related to one another there in the second part of the spike protein. Why isn't this going backwards? There we go. And anyway, so when SARS-CoV-2 and these are compared on the amino acid level, the S2 portion of the spike protein is 95% identical. The ORF8 protein is 94%, and the E protein is 100% identical. The E protein's 100% identical sequence was also identified in the Nerd Has Power blog, and was also later explored in terms of the evolutionary relationship with other coronaviruses by Daewoo, this now legendary molecular biologist on Twitter. Anyway, the point is that no other coronaviruses share more than 58% identity with this particular protein, and that's the ORF8 protein. And the ORF8 protein is very important for invasion. And you remember all the things that we've discussed before. The laboratories in China were involved in years and years of sampling coronaviruses to find different variants of all of these proteins that they use for replication and that they use for hijacking the host cell machinery. I've shown you papers where they were looking at ORF6, ORF8, ORF1. They're talking about open reading frames which contain proteins within them. So in this case, ORF8 could have a couple different proteins that it codes for. ORF1 will have many different proteins that it codes for. So all of these open reading frames are just things that in the genome of coronaviruses that the Wuhan Institute and American institutes and Australian institutes and institutes all around the world that are interested in coronaviruses have been studying as units because they're easily characterized and easily moved from virus to virus functionally. That's the whole point. So comparing the this is the spike protein here, and comparing the S1 and the S2, you can't really see that in this picture. But their point is, again, these are the open reading frame 1A and open reading frame 1B. These are the two giant portions of the genome that contain many proteins that are vital for the replication of the virus. I think ORF1B is the one that contains the RNA that copies the RNA of the virus. Anyway, we'll go on. The E protein is a structural protein which is embedded in and lines the interior of the membrane envelope of the viron, and the E protein is tolerant of mutations, has evidence in both SARS and related bat coronaviruses. Let's see if we can go on to this figure 2b. So here what it's looking at is in all of these different proteins, in these E protein sequences, there's a lot of tolerance for different mutations. And the reason why is because the E protein doesn't really do very much. If you change its shape a little bit, it's okay. It doesn't really change much for the effectiveness of the virus, okay? And that's why it's not under a lot of evolutionary pressure. Therefore, it's very strange that it wouldn't be altered. And the E proteins of SARS-CoV-2 have already undergone mutational change. Sequence data obtained during the month of April reveals that mutations have occurred at 4 different locations in different strains, and that's figure 2C. Consistent with this finding, sequence blast analysis indicates that with the exception of SARS-CoV-2, no known coronaviruses share 100% amino acid sequence identity on the E protein with ZZ45. Although 100% identity on the E protein has been observed between SARS-CoV and certain SARS-related bat coronaviruses, none of those pairs simultaneously share over 83% of identity on the ORF8 protein. So again, what they're trying to show you is that the combination of this high homologies across the functional domains of the virus, that's the significance of it, because the homology is and lack of homology is organized very well around functional domains of the virus, and random mutations and random evolution, generally speaking, doesn't generate that type of pattern. That was that picture. You might have to read that for yourself a little better to look at how these sequences are from recent coronavirus. So the bottom one here, you can see how long the sequence has been stable here. So if you look here, the bat coronaviruses that we're interested, ZZ45, that's the top line here. And then if you look at these ones here are all new coronaviruses taken at different times, February, April, different places. And then they're looking at how little, it's not very many changes, right? There's a little change here, there's a little change there, and these black ones are changed. But it's very, very, very few changes from bat ZC45 and this ZXC21. And these viruses were reported in like 2013 or 2014 when they were found, and then again in 2017 when they were discussed. So we're really talking about some weird stuff here. It's not very likely that this virus was around for 3 or 4 years in the wild. These viruses can't be sampled again. These are viruses from the past. The receptor binding motif of SARS-CoV-2 spike cannot be born from nature and should have been created through genetic engineering. So, so far what they're contending is that I think I went past this one. Sorry, I read too quickly. Just reading their own text here so you can kind of just follow along on their logic. Importantly, ZC45 and ZXC21 are bat coronaviruses that were discovered between July 2015 and February 2017, isolated and characterized by the military laboratories. They were published in 2018. Clearly, this backbone template, which is essential for the creation of SARS-CoV-2, exists in these and other related research laboratories. What strengthens our contention further is the published RATG13 virus. So here they're going to do what we have done in the past with our discussions and look at how RATG13 is not really, it doesn't really make sense that this is a real virus, because a very recent publication, as you can read down here, a very recent publication has also indicated that the receptor binding domain of RATG13 spike protein could not bind the ACE2 of two different types of horseshoe bats. So what does that mean? What is this sentence right here, this very recent publication? And it is a very recent publication. What this means is that, I'm just going to scribble on here so you see where I'm going. What this means is that you can't get a bat virus out of a bat if it doesn't have a way of infecting the bat. And so it physically couldn't be a bat virus if it can infect the bat that you said it came from. The other thing that's really interesting is that bats have a very high body temperature. And so it's not very clear that RATG13 as a viral sequence could even survive at the high temperature of the bat. So it's all, there's a lot of questions here, and of course I'm very, very much blowing through this in that sense, right? I'm not being totally fair with this. They're also they go a lot more in detail. They bring up Nature articles that bring up this conflict between the RATG13 sequence and ZC45 that was quickly retracted for rectification and never came back out. So they've done a very good job in this paper of also characterizing and not characterizing, but cataloging the events that have gone along with this. So in the second part of this paper, what they're going to look at is the receptor binding motif, and specifically the fact that it couldn't have come from nature but must have come through genetic engineering. And so the high sequence identity, again, is specific of in SARS-CoV-2 is specific for the S2 portion of the spike protein, and the S1 portion of the spike protein is the portion of the spike protein that actually is binding to ACE2. And so the convenient sort of, that ain't even convenient. It's like convenient for us because S2, if it doesn't have much functionality in terms of host specificity, then there isn't a lot of evolutionary pressure on it to stay very stable, and yet it's incredibly 195% similar to the S2 of these two guys. So that's an issue, again, that seems to indicate that there's something wrong here. And then the other problem, of course, is this furin binding or furin cleavage site in between. So just to give you an idea, in case and you hadn't seen this before, the S1, I'm sorry, go back one. The S1 protein and S2 proteins of the spike protein go together in a dimer. I don't even know if they call it a dimer, but they go together like this. And then 3 S1, S2 units form a trimer. So they get together in this little sequence like this. And so if you look at it from the bottom and you flip it over, it looks like this, right? And so now these S1s are down here, and then when they get cleaved, they can hang out. And so that's what the cleaving is all about. The cleaving is sort of opening up this trimer so that these things are a little bit more flexible, and the S1 can more easily access the ACE2 protein, and here's where you see that here. What they're showing you here now are the amino acids which are important for the interaction between S1 and ACE2, and then they're going to make the argument later on in the paper that all of these essential amino acids, the amino acids that are essential for binding to ACE2 from a structural perspective, are preserved somewhat serendipitously in the sequence of SARS-CoV-2 from the original SARS, too serendipitously by these authors' estimation. Similar to what is observed for other viral proteins, S2 of SARS-CoV-2 shares a high sequence identity with SARS. Yes, in stark contrast between SARS-CoV-2 and this SC45, ZC45, I'm never going to be able to say that. The S1 protein, which dictates most host virus can infect, is much less conserved, with the amino acid sequence identity being only 69%. Okay, so figure 4 shows the sequence alignment of the spike proteins for 6 coronaviruses. 2 of the viruses are isolated from the current pandemic, 2 are suspected template viruses, and 2 are SARS coronaviruses. The receptor binding motif is highlighted in between the 2 orange lines. Clearly, despite the high sequence identity for the overall genomes, the receptor binding motif of SARS-CoV-2 differs significantly from those of the other 2. So let's go look quick to check that. So this is the whole sequence here, and we're going to zoom in on this part. This is the whole sequence right here. And I believe, let's go back again, and says clearly yes, 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 although yes, highlighted between the 2 orange lines. So this is the receptor binding domain, or the receptor binding motif. The red lines mark the crucial amino acids that they believe are responsible for, or most responsible for, the physical interaction between the virus and ACE2. And these, they note, are also the ones that are not changed. So let's see what we got here. This is Wuhan. This is SARS-CoV from America. These are the 2 bat viruses we're interested in. And then these 2 on the bottom are the SARS from 2003. Okay. So let's zoom into this here. And so again, the 2 SARS-CoV-2s are on the top. The suspected template viruses are in the middle here. And then the original SARS-1 is here on the last 2 lines, and then a consensus on the bottom. So if we look at all these red lines, what we're looking at are amino acids that these authors claim are the amino acids that are depicted here in the orange sticks. So all of these orange amino acids here are responsible for this interaction between S1 and ACE2, and these are the amino acids that are here in the entire sequence. And this, again, remember, is the sequence of just the S protein. The entire virus is about 30,000 base pairs long, or 30,000 amino acids long. This is only the spike protein. And so what we're looking at here, this is, I believe, the furin cleavage site here in green, and then this is the part that interacts, of the spike protein that interacts with the ACE1 receptor. So I'm going to go back here and just make sure we didn't miss anything on this part. Yes, so they're talking about some of the things that are changed, and the few changes within the group of essential residues are almost exclusively hydrophobic substitutions. And so what they mean there is that when you look at a protein like this, what is here is basically charged or uncharged amino acids. And if you substitute amino acids with similar properties, either they like water or they dislike water in this case, then amino acids aren't that different from one another in general if you maintain their general classification and their general charge. And in this case, what they're suggesting is that since any of the changes that have happened are almost exclusively hydrophobic substitutions, meaning one hydrophobic for another, it won't really affect the protein folding, and therefore might not affect the interaction with human ACE2. In fact, it's possible that they could even help. And recent structural work has confirmed this for them. So as elaborated below, the way that SARS-CoV-2 receptor binding motif resembles SARS-CoV receptor binding. So now they're making the argument here, and it's you got to make sure you follow this. They're now arguing that the receptor binding motif of SARS-CoV-2 resembles that of the original SARS. I think this is kind of a relatively novel claim from this paper. It's one of the things that none of us were really discussing in great detail. But I think with their analysis, it's very neat that they have come up with this. So what they're saying is that what we're seeing here is a selection for this receptor binding motif, and then they worked on it basically. The virus would have had to adapt exclusively to its new host. And so what they're talking about now, if SARS-CoV-2 does indeed come from natural evolution, its receptor binding motif could have only been acquired in one of two possible routes: an ancient recombination event or a natural recombination event that occurred relatively fairly recently. And that's not very likely, because that would have been very random and had to have inserted this cleavage site and all this other things. So that's not very likely. But in the first scenario, the ancestor of SARS-CoV-2 would have recombined and swapped its receptor binding domain with a coronavirus carrying a relatively complete receptor binding motif in reference to SARS. This recombination, right, it would have had to have been already very close to where we are now. Otherwise, the recombination would have been pointless. So let me see if I have figure 4 here. No, I don't. I don't know why I don't have figure 4. I know, but because figure 4 was back there. So with all of the gaps in its receptor binding membrane motif filled, subsequently the virus would have had to adapt extensively to its new host. And this is the kind of thing that we've been talking about for a long time. If it comes out of a bat and it goes to a human using the same ACE2 receptor, it's impossible for it to be well adapted to the bat and then jump to the human and be immediately well adapted. It has to adapt a bit during this jump. That's the whole point of zoonosis. That's what Alina Chan shows in her, preprint that's been out there for like 3 months now that nobody will publish because it doesn't go with their narrative. Random mutations across the genome would have had to have occurred to eventually shape the receptor binding motif to its current form, resembling SARS-CoV receptor binding motif in a highly intelligent manner. However, this convergent evolution process would also result in the accumulation of a large amount of mutations in other parts of the genome, rendering the overall sequence identity relatively low. The high sequence identity between SARS-CoV-2 and ZC45 on various proteins do not support this scenario. And that's what we've been talking about before. If it's a crossover event, then the crossover event defines the receptor binding motif. But then by definition, all of the other changes have to have another, or all of the other consistencies have to have another explanation. And that's really the problem. And so that's where they're going with this argument. In the second scenario, ZC45, like coronavirus, would have had to have recently recombined and swapped its receptor binding motif with another coronavirus that had successfully adapted to bind an animal with ACE2 highly homologous to human ACE2. So this is where ZZ45 would have recombined with something like a pangolin. This is that other story, right? That the two different viruses then have to share significant sequence similarity, and they must also co-infect and be present in the same cell of the same animal. And that the recombinant virus would not be cleared by the host or make the host extinct. That the recombinant virus would eventually have to become stable and transmissible when the host species. So if you read that long list of requirements for this to work, you really get an understanding of why even the story about the original SARS in 2003 is not very biologically plausible, and why it's become under question again by a lot of us and people at the time also, but a lot of us are reopening that box as well. Let's read that again. The likelihood of such events depends in part on the general requirements of natural recombination, which are, 1, that the two different viruses share significant sequence similarity, that they must co-infect and be present in the exact same cell of the same animal, right? Because that's the only way that they can exchange genetic components, and that the recombinant virus would not be cleared by the host, meaning that it has to escape. And it also has to not kill the host before it can pass on. And that recombinant virus would eventually have to become stable and transmissible within that host species. So these things are really crazy, and very nicely put forth, and very worth your time to take to read again, because your brain will start to really understand how absurd the natural zoonotic story is, and why people like Peter Daszak really need to check themselves, because we're coming for them. In regard to this recent recombination scenario, the animal reservoir could not be bats, because the ACE2 proteins bat is not homologous enough to human ACE2. So that's the reason why we need a host between bats for this virus. That's the reason why we've always argued that we need a host. That's why there had to be a market. That's why there was a civet. That's why there's been snakes. That's why there were pangolins. You see, this is the thing, and they're breaking down for you the biological implausibility of this. Excuse me for a second. So, in addition, there has been no evidence of any SARS-CoV-2 or SARS-CoV-2-like virus circulating in the human population prior to late 2019. And intriguingly, according to a recent bioinformatics study, SARS-CoV-2 was well adapted for humans at the start of the outbreak. So anyway, we are looking at basically they took a virus and they made it. The only other possibility of natural evolution remains, which is that ZC45, a like virus, and a coronavirus containing a SARS-like receptor binding membrane motif could have recombined in an intermediate host where the ACE2 is homologous. So this is kind of the same thing, and I guess they're going to go, yeah, pangolins, here you go. So sorry, shouldn't have said this. I should have read this to you. But this is a detailed thing here. But remember, they also point out that these findings have also been recanted, right? There's no coronaviruses in some of these pangolins anymore. And an in silico study, they're citing all of the things. The in silico study that they're citing here, as I point to it with my finger that you can't see, the in silico citing that they're pointing here is this study from the Australian fella, where he looked at the binding affinity to different ACE receptors and showed ACE2 receptors and showed how ridiculously off the charts it was for humans. As we have described above, if natural recombination events is responsible for the appearance of SARS-CoV-2, then the ZZ45-like virus and a coronavirus containing would have had to recombine in the same cell by swapping the S1 receptor binding motif, which is a rare form of recombination. Furthermore, since SARS has occurred only once in human history, it would be at least equally rare for nature to produce a virus that resembles SARS in such an intelligent manner, having a receptor binding motif that differs from the SARS receptor binding motif only at a few non-essential sites. The possibility that this unique SARS-like coronavirus would reside in the same cell with ZC45-like ancestor virus with two viruses would recombine the receptor binding motif swapping fashion is extremely low. Importantly, this and the other recombination event described below in sections 1.3, even more impossible to occur in nature, would both have to happen to produce a spike as seen in SARS-CoV-2. So what they're saying here, just to be sure you get it, is that they have even more things to discuss below, and what they're going to discuss below is even more impossible to occur in nature than what we just discussed. And so that's a pretty big deal. While the above evidence and analysis indicate together that appear to disprove the natural origin of SARS-CoV-2's receptor binding motif, abundant literature shows that gain-of-function research where the spike protein of a coronavirus was specifically engineered has repeatedly led to the successful generation of a human-infecting coronavirus from coronaviruses of non-human origin. So what are they saying there? Well, we've seen it many times that they can pull viruses out of nature and they can make these viruses work, right? They've shown us this many times, and so there's no reason to doubt they couldn't do it. Records also show that the research laboratories, for example, the Wuhan Institute of Virology, have successfully carried out such studies working with U.S. researchers and also working alone. In addition, the WIV has engaged in decades-long coronavirus surveillance studies, as I just told you, and therefore owns the world's largest collection of coronaviruses, which the database that was the catalog for them was conveniently deleted earlier this year. All of them. First they changed all of the headings and then they just deleted the whole database, all 4 of them. Evidently, the technical barrier is non-existent for the WIV and other related laboratories to carry out and succeed in such spike receptor binding motif engineering and gain-of-function research. Now, this is where it gets very good, and this is where I might need to insert a little part here where we talk a little bit. Strikingly consistent with the receptor binding motif engineering theory, we have identified 2 unique restriction sites, EcoR1 and BSTE2, at either end of the receptor binding motif of the SARS-CoV-2 genome, respectively. What they are showing you here is a sequence of amino acids and a specific sequence of bases where a specific restriction enzyme cuts. And why is this important? Maybe you don't know what a restriction enzyme is. But molecular biologists have basically gone through nature, bacteria, fungus, eukaryotes, whatever, looking for restriction enzymes. And what restriction enzymes are enzymes that cut DNA and nick DNA in very specific locations that are base pair and base sequence specific. EcoR1 only cuts there. It only cuts when it finds a GAATTC, and it only cuts there. And it looks through any other genome. It won't cut anywhere else. It won't ever make a mistake. It only cuts there. Here's a BSTE2 site, which is GGTTACC. Anywhere where this is, this enzyme will nick that DNA, cut it there. Why is this important? Why is this cool? Why is this interesting? Where did my pen go? The reason why this is interesting is because it can be used as a tool. What happens is that when you cut with an enzyme like this, it makes a very specific cut. Let's say it does it like this. What that means from a DNA perspective is that you've created, sorry, I'm going to do that again. We're going to go back like this. I'm just going to make a separate picture here. What it does is it makes like a little edge that looks like this. And the other edge looks like this. Now, the point is that these things are complementary shapes. And the interesting thing about this is what they're going to imply is that the fact that there's a cutter that can make a cut with a specific shape. So imagine this shape is that shape. This one cuts that shape. And so what they're implying here is that because SARS-CoV-2 has restriction sites, very specific restriction sites here and a restriction site here, what it allows you to do is remove this part in between and swap it with another part as long as what you're pulling out from the other place has these 2 restriction enzymes on either end of it. The basic gist of it is that if you have a sequence like this, and it's a long line, and you cut it here and you cut it here, you cut it here with the B enzyme and you cut it here with the E enzyme, right? Then you get this piece. You can take another enzyme, another piece of DNA, and you can cut it with the B enzyme and the E enzyme. And this piece will fall out, right? And then what you'll have is a hole here, this hole, will have an end with an E and an end with a B, right? And then this piece is missing in the middle. But this piece right here has the complementary B and E ends that allow you to just take it and drop it right in here into the hole you just cut. This is one of the worst molecular biology diagrams ever drawn on YouTube, and the let me check. The 27 of you that are watching this video right now just witnessed the creation of the worst molecular biology illustration of what a restriction enzyme does ever created on YouTube. But if you followed it, that's really great. The point is this enzyme can cut a very specific shaped end that's complementary. It's like a male and female end specific for this enzyme target site. And this makes a male and female end specific for this target cut site. That means that if you take this part out of the middle, it'll have an end here and an end here, which would be compatible with any other place that you cut a hole with this enzyme and this enzyme. And so what they're going to imply here, you know what I want to do? What I really want to do is I'm going to go over here and I'm going to end the show quick, just quick. And I'm going to add a thing there. Then I'm going to start the show up again. Yeah. And then I'm going to go here and I'm going to draw something for you. See, here's the thing. What we got is you got this sequence of DNA. And we want to take this little piece out here. This is the piece you want to take out. And you want to swap that with this. This is the other viral genome over here. And it's got its own receptor binding motif. And what these authors are implying is that where is this pen color? Yeah. What they are implying is that the original ZC45 has no restriction enzyme sites there, but they were conveniently close. It was only a couple amino acids that needed to be changed before there would be two very convenient restriction enzyme sites there that would allow them to cut at this location and cut at this location and end up, and this is the thing, they would end up with two ends right here that were compatible. These two ends would be compatible with any other place, any other sequence where you cut with those same two enzymes here. So if you could cut this receptor binding domain out with the same two enzymes, then by mixing these two in the same cell, you can get them to swap. And in fact, what's very cool is, because we've talked about this before, because you can do this so easily with just making a circular DNA and letting a cell line make the virus for you. You can design it from scratch. They only needed to do this from the perspective of sampling new spike receptor binding motifs. So it was easier, in fact, than taking the whole spike protein, because you could preserve the spike protein itself and only take this interesting part and screen that. And that's what's so spectacular about finding these restriction enzyme sites, is because they are essentially a smoking gun for a Lego type set for viruses. You could just use it like Legos. You could just, whoa, what was that? Oh yeah. You could just use it like Legos. You could just move this piece out of one virus and put it in another one, as long as you cut both ends with the right scissors, because they would leave the right shape so that it would fit nicely in to the part that you would cut out of the other virus. And so this makes it really cool. I'm going to go back back here. I just want you to be able to see everything as best as possible. Also, although it may be convenient to copy the exact sequence of the SARS receptor binding motif, it would be too clear of a sign of artificial design and manipulation. The more deceiving approach would be to change a few non-essential residues while preserving the ones critical for binding. And so that's what they're telling you, and that's what I told you before. If they just looked at the high-resolution structures, like I showed you in the previous picture, that was figure 3 with all of the sticks and the trimer and that kind of thing. We believe all of the crucial residues, and those are labeled by the red hashes in figure 4 that I showed you again. They should have been kept, and as described earlier, while some should be direct preservation, some should have been switched to residues with similar properties, which have not disrupt the human ACE2 binding and may even strengthen the association further. And again, with a little work, they could have screened for this. And that's what these guys are implying here. Okay, just very quickly about the furin cleavage site. They're going to go into more detail than I do, but just to hit this point home, the furin cleavage site is in between the S1 and the S2 portions of this protein. The S1 is the novel portion. The S2 has a very high homology to our suspicious ZZ45. And in contrast, the furin cleavage site at this location has been observed in other groups of coronaviruses, but not in beta coronaviruses. And certain selective pressure seems to be in place that prevents the lineage B of beta coronaviruses from acquiring or maintaining such a site in nature. And that's the evidence that the cleavage site disappears whenever you put it in other animals. It doesn't survive. It just gets eliminated right away. This is a picture of a vast list of SARS coronaviruses, SARS-related coronaviruses, and this is the 4 amino acid insertion in SARS-CoV-2 that just doesn't simply line up with any of the other viruses that we have in task. And that's really key here, because remember that one of the arguments that was made early on was simply a misalignment, where they shifted some of these things and then made it seem like that this wasn't really an addition of 4 amino acids novelly, but it is. This furin cleavage site requires the addition of 4 amino acids, which are simply not present in any other virus. And that's a real problem from an evolutionary perspective, because that's not how these things work. Consistent with this claim, a close examination of the nucleotide sequence of the furin cleavage site in SARS-CoV-2 spike has revealed that the 2 consecutive arginine residues within the inserted sequence are both coded by a rare codon CGG, the least used codon for arginine in SARS-CoV-2. In fact, this CGG-CGG arrangement is the only instance found in the SARS-CoV-2 genome where this rare codon is used in tandem. This observation strongly suggests that this furin cleavage site should be a result of genetic engineering. Adding to this suspicion, a FAU1 FAU1 restriction site is formulated by the codon choices here, suggesting the possibility that the restriction fragment length polymorphism, and that English is not very good, so let me read that again for you. This is a really key thing for me to help you understand, I think. Adding to this suspicion, a FAU1 restriction site is formulated by the codon choices here, suggesting the possibility that the restriction fragment length polymorphism, a technique that at the WIV lab is proficient at, could have been involved. So restriction fragment length polymorphism is a technique for tracking mutations in DNA sequences. I think I'm going to try and go back to this previous this thing here. So remember what I told you before about what a restriction enzyme is. A restriction enzyme will take your DNA sequence and it will look for a very specific set of sequences of bases, and it will cut that DNA sequence in such a way as to leave complementary ends like this. And so you get this, these are not arrows. These are just kind of things to indicate what's going on here. So you get a complementary sort of looking thing, right? You got a hook on this end, hook on that end, hook on this end, hook on this end. And so you can imagine a scenario where this part, just work with me here. I know this is a terrible drawing. That this part can't join up with just any shape, but it needs to join up again with this kind of a shape. I'm just going to go back and clear this again. So the point I'm trying to make is that restriction enzymes do that. What they do, remember what we said before, they look for a specific sequence, they make a cut, and then they go, and they find another sequence, and then they cut it, and they're going to go, and it goes all through the genome, finds another one, it cuts, it goes all the way down. That's what a restriction enzyme will just do. That if you put it in an Eppendorf tube, it'll just go through a whole sequence of DNA and it will cut it at every place where it finds the sequence it's interested in. Okay? What a restriction fragment digest is when you take an enzyme, let's say there was an enzyme called JC1, and JC1 cut at all these circles. If you ran the products of this cuts, right, because now you have a product that's this long. After the cuts, you have a product that's this long. You have this really long thing here, like this whole thing is one product, right? And you have this really long thing here. You have this long thing that goes all the way down here. So it's 1, 2, 3, 4, 5, and then a little one right here is 6 fragments. And so if you ran this on a gel, what you would see down the gel are lines for the lightest and heaviest fragments. So the 6 fragments would all sort out like this, and they would show up as lines on your gel. And the smallest fragment, which is the one down here at the bottom, this one, would show up way down here because it would run the farthest on the gel because it's the lightest one. It goes the farthest. And so what you'd be able to do then is look at these fragments and say, okay, there's 1, there's 2, there's 3, there's 4, and you'd get a weight for each one of them. And if you looked at another set of DNA and one of these restriction sites was missing, then one of these fragments would disappear from your gel, and you would immediately know that, I don't have the same protein anymore, just from this one restriction digest. And so now imagine a scenario where we took this whole protein that we're interested in. Here's our sequence, right? And we did a restriction digest of one enzyme, and you get 3 fragments. And then we do a restriction enzyme with a digest with another enzyme, and we get, let's say, 5 fragments. And then we do another restriction enzyme digest with another, and we get only 2 fragments. So now if we do a restriction digest on this sequence, which is the blue line, with 3 different enzymes, so we're going to do it in sequence, right? We're going to first do it with the one enzyme, and we're going to get 3 different fragments. Then we're going to do it with the second enzyme. We're going to get 1 fragment, 2 fragments, 3, 4, 5 fragments. 1, 2, 3, 4, 5. And then we're going to do it with the X, and that enzyme only cuts it once, so we end up with 2 fragments that are quite heavy. So this single line, this long line here, could be characterized by a restriction digest with this enzyme, and this enzyme. And if you did that, what you would get is for this enzyme, you would get 3 fragments. For this enzyme, you would get 5 fragments. And for this enzyme, you'd get 2 fragments. And they would be predictable weights, right? Because this is the weights going down this way. And so if this is the weights going down this way, wow, that's a terrible arrow. Then what you have are, with 3 restriction enzymes, you get 3 predictable, 5 predictable, 2 predictable fragments, and you can identify this protein. And more importantly, if this protein changes at any of these restriction sites that might be interesting, then these fragments will change in size and some of them will disappear. And so you can physically track. Wow, that's awful. When I look at the stream, I'm ashamed. You can physically track whether or not you lose this inserted sequence. And so very specifically, keep in mind, again, right, what they're saying here. This inserts a FAW1 restriction site, which allows them to follow it, because if this drops out, if this CGG, CGG, GCA, if this sequence drops out, whoops, damn it, sorry for that. If this sequence drops out during any of their passage or any of their laboratory work, they can just do a quick restriction enzyme, a restriction digest, and see if this enzyme still cuts here. Because if it doesn't cut here, then the chances are very good they lost this crucial insertion. And so the choice of these very strange arrangement here, these authors are suggesting, is a choice which belies the desire to insert a FAW1 cut site, a restriction site, so that they could follow it and not lose it. It's not the perfect explanation, but I think you should be able to follow that, because if this sequence changes, then this enzyme won't cut there anymore, and they can see that when they run it on a gel, because they'll be missing a fragment, or a fragment will have changed to a different size. And that's what restriction fragment length polymorphism is. Polymorphism is the changing of this of things, the multiple forms of things. So restriction fragment length multiple formism, right? So if you cut with a restriction enzyme and the fragment lengths change, then you get this indication that you lost this thing. So it's a really interesting and totally exciting indication in my mind that this could be genetically engineered. And it's a nice observation that I hadn't heard before specifically. So I'm just going to read this summary, and then I'm going to give you some more crazy stuff. Evidence presented in this part reveals that certain aspects of the SARS-CoV-2 genome are extremely difficult to reconcile to being a result of natural evolution. The alternate theory we suggest is that the virus may have been created by using this as a backbone, and this is supported by the finding of unique restriction enzyme digestion site at either end of the receptor binding motif. And that was, again, so that they could pull the receptor binding motif in and out of different viral constructs and test how functionality moved. An unusual furin cleavage site may have been introduced and inserted at the S1-S2 junction, which contributes to the increased virulence and pathogenicity of the virus. And this was where, again, that FAW1 cleavage site was inserted as well. At least they suggested it might have been, so that they could follow and make sure that they didn't lose the unusual furin cleavage site through animal passage. These transformations have then staged the SARS-CoV-2 virus to eventually become a highly transmissible, onset hidden, lethal, sequelae unclear, and massively disruptive pathogen. Evidently, the possibility that SARS-CoV-2 could have been created through gain-of-function manipulations at the WIV is significant and should be investigated thoroughly and independently. You could slam the door right now, and this paper is awesome. And they take it further. It's just spectacular. What they're going to show you is that there are 5 basic steps to make this virus. First, you need to select the SARS-CoV-2 backbone that you want to use. It's like selecting your basic t-shirt model. Then you need to engineer a spike protein to bind ACE2. And what this means is that you can't just put SARS on it. You can't use the old SARS, because that won't work. You have to make one that's better. And so that's what they've done, and we've seen lots of papers that do that. Then you've got to select the right ORF1b gene. That's the open reading frame 1b gene, which also contains this RNA-dependent RNA polymerase. That's the RDRP protein, which they always call RDRP, but it's actually the most important protein in the viral genome. It is the RNA-dependent, so RNA-dependent RNA, RNA polymerase. Hello. RNA-dependent RNA polymerase. That's what that is. That's the main thing that replicates the genome and gets the genome of the virus going, gets it replicated, and makes more virus. They have sequenced this particular protein on every virus they've ever cataloged. Shang-Chi Li's primary modus operandi is to sequence every single virus's RDRP and just call it an RDRP instead of calling it the most important gene in the genome. Then you need to use reverse genetics to assemble it into a cDNA clone. If you remember from my bike rides and from many of my other discussions, I have told you many times that this cDNA clone technology of making RNA viruses was perfected between the years of 1998 and about 2005. And by the year 2003 already, Ralph Barrick's lab had resurrected SARS-CoV viruses from 2003 that were extinct, brought them back from a text sequence to a live virus in the laboratory. So that's what they're talking about here with number 4. And then number 5 is serial passage in vivo, and that's to clean up the mess, to make sure that everything works right, to make sure that the virus works well in vivo and is a little bit stable before you let it go. And so these 5 steps are outlined here. It's interesting because they're also citing a lot of the papers that I've discussed on previous bike rides. In the second part of this report, we describe a synthetic route for creating SARS-CoV-2 in a laboratory setting. It is postulated based on substantial literature support, as well as genetic evidence present in the SARS-CoV-2 genome. Although steps presented here should not be viewed as exactly those taken, we believe the key processes should not be much different. Importantly, our work here should serve as a demonstration of how SARS-CoV-2 can be designed and created conveniently in research laboratories by following proven concepts and well-established techniques. This is the argument that I've been making from the bicycle for several months now. I'm sure most of you guys who are still here with me tonight are aware of that, so I don't need to really push that. But in case you're not aware, there are several bike rides from the previous months which have discussed these techniques and made the argument that there's no reason to believe we couldn't have used these techniques to have created and established this. So the possible scheme in designing the laboratory creation of the virus. First, you need to engineer or create a human targeting coronavirus. They have to pick a bat coronavirus as the template and backbone. And this can be done conveniently because they have been collecting bat coronaviruses over the past 2 decades. This is a point that I have made, and I think they make it very succinctly here. The template is ideal, but the template shouldn't come from Dr. Shang-Chi's collections because she's widely known to have engaged in the gain-of-function studies. Therefore, these 2 viruses are suitable because they come from this military laboratory and in general haven't been worked on. So that's an interesting postulate that I hadn't really thought of before and hadn't seen this as a reasonable reason why ZC45 would be an especially good backbone, but I get their argument. The next thing you need to do is engineer through molecular cloning a spike protein that can bind human ACE2. And this isn't a problem because, again, they've shown that they can do this in the literature. They have gone through swapping spike proteins, and they've also gone through swapping receptor binding motifs. So this is no problem. Second, they would use molecular cloning to introduce a furin cleavage site, and this manipulation based on known knowledge would likely produce a strain of coronavirus that is more infectious and pathogenic. What they mean here is that they've seen the gain of function that a furin cleavage site gives other viruses in the past, and so it wouldn't be a ridiculous thing for them to try and set out to make a furin cleavage site in a coronavirus spike protein without knowing for sure if it would make things better. But they could assume that it would, and it wouldn't be a crazy assumption based on several papers from the past. So now we're at this third stage here, the ORF1b gene that contains the RNA-dependent RNA polymerase, RDRP. So remember, among them, the RDRP protein is the most crucial one and is highly conserved among coronaviruses, and importantly, Dr. Shang-Li Shi's laboratory uses a PCR protocol which amplifies this particular fragment of the RDRP gene as their primary method to detect the presence of coronaviruses in raw samples. As a result of this practice, their group has documented the sequence information of this short segment of the RNA-dependent RNA polymerase for all coronaviruses that they have successfully detected or collected. This is a very serious point here, because what this means from my perspective is that they have more than enough intellectual property in their databases that they deleted in order to have selected the best RNA-dependent RNA polymerase, the best helicases, the best methyl transferases, and the best endoribonucleases. These selections were most certainly made. These screening sequences, the screening of these different proteins in different coronaviruses was most certainly done. And so selecting this gene was definitely something that they would have had to do. And here, the genetic manipulation is less demanding or complicated because the ORF1b is conserved, and likely any beta coronavirus would be competent enough to do the work. However, we believe that they would want to introduce a particular ORF1b into the virus for one of two possible reasons. This is interesting. Since many phylogenetic analysis categorize coronaviruses based on the sequence similarity of this RDRP gene, having a different RDRP gene in the genome, therefore, would ensure that SARS-CoV-2 and ZC45 are separated into different groups of phylogenetic studies. Choosing an RDRP gene, however, is convenient because the short RDRP sequence has been recorded for all coronaviruses ever collected or detected. Their final choice was the RDRP sequence from the coronavirus 4991, which was discovered in 2013. For 4991, the only information ever published was the sequence of its short sequence. So this is where I think they really take it to a new level, this part right here, because this part is where we have been thinking that this virus, 4991, is the one that's probably RATG13 and then plus some keyboard stuff on this thing. These guys have analyzed it a bit further and would suggest that actually they selected this specific short segment from this virus. And so I find that I'm going to go back and forth here just to get back on with a no pen. After amplifying it, they would then use it for subsequent assembly and creation of this SARS-CoV-2. So now you see why it's so cool. This paper has really put together the pieces in a way that we hadn't been able to do before, that no one had done before. So what we have is a RNA-dependent RNA polymerase that is taken from a virus discovered in 2013. That virus is definitely from the caves in the Moyang province. The ZC45 virus is also a virus that may have come from the same collection period, same collection area. But according to this report, this one was found later than the 4991 virus. And so this is cool again because, right, we're talking about trying to choose a RNA-dependent RNA polymerase so that by definition, when classified, the wouldn't meet in the phylogenetic analysis because this virus and this virus are going to get classified in different phylogenetic lineages. And so it makes them look like this one is not related and that one's not related. It's a clever observation. And so then you fabricate this sequence, and then this sequence throws you off. You don't get from RATG13 to ZZ45 because of the fact that 4991 exists. And that's where Mona probably, she doesn't trip up. Mona didn't make a mistake. Mona's paper is great. But the connection sequence here is because primarily this sequence is so short. And this sequence is probably the one that they use. So I really like this argument. I don't know if it's one, a chicken comes before the egg or whatever this is, but I do think this ties a little bit of the loose ends up for me. and starts to make more sense from the perspective of where these different things come from. So number 2, I got to look down here and see what this is. So then the RNA dependent RNA polymerase protein from this 4991 is unique in that it is superior to them from any other. And so that's the other thing that I find very cool about this, is that they're going to tell you now the other reason why they might do this. Go back. Oh, please help. The other reason why they would do this is because in this part, number 2 here. Oh my goodness, that's terrible. But number 2 here is really cool because this is the other thing that we've been talking about. If you find that this protein is the most important protein for viral replication and the most important protein for, it's the conserved protein, right? It's conserved across CoVs. What does it mean to be conserved across CoVs? That means that this protein is so important that it doesn't change. This is a very, very, very important point for us all to understand very well. Okay. What this means is that this protein is so important that it can't change. It doesn't change. It has been honed by evolution. And so now what they say in number 2 here is, imagine a scenario where the RNA-dependent RNA polymerase from 4991 is unique in that it's very, very stable and very, very superior in some way. Reliable, fidelitous, doesn't make very many errors. This is important because if it makes errors, then a virus doesn't stay virulent. If they found a very superior RNA-dependent RNA polymerase that was very not error-prone, it would be a very serious finding. It might be one of the reasons why they are always screening for this protein. And if that's the case, and they found one that was very good, then you know what we got here? We got a wonderful antiviral target. And in fact, remdesivir targets this protein. Are you kidding me? Are you kidding me? How is this lining up for you now, folks? Are you enjoying this? This is fantastic. Because essentially what we have here is an explanation. It's a smoking gun. It tells you what they were doing. They were screening different coronaviruses for very fidelitous, that means RNA polymerases that don't make mistakes, and that is a very rare thing in nature. I can tell you that as a biologist. RNA polymerases make mistakes. DNA polymerases, much more high fidelity, much less error prone, and there are many more enzymes that are looking for those errors to correct. This is the smoking gun, because what they were doing was screening viruses for the most effective RNA-dependent RNA polymerase, and this is one of the reasons why SARS-CoV-2 would be so stable in such a large population of infections, because its RNA-dependent RNA polymerase is so stable. It was selected from a screening of decades of coronaviruses. We're really on to something here, folks. This is a really important paper. So fourth, they would use reverse genetics to assemble the gene fragments of the spike and the rest of the template into a cDNA version of the viral genome. They would then carry out an in vitro transcription to obtain a viral RNA genome. Transfection of the RNA genome into cells would allow recovery of live and infectious viruses of the desired artificial genome. That would be like viro cells. And then they take those viro cells out and they would put they take those viruses out and they would put them into an animal. It could have been monkeys. It could have been these golden hamsters. It could have been ferrets. Any animal with high homology with our human ACE2 receptor would have been appropriate for the 5th step. And the 5th step is also, of course, the one that really cleans it up and makes it impossible for anybody to really see the signs of any manipulation. It just smooths the edges over a little bit. But these authors have really seen through it, I think, quite spectacularly. So again, you select a CoV backbone, you engineer the spike protein to bind ACE2, then you have to select this ORF1b gene, and specifically you want to get the best RNA-dependent RNA polymerase you can buy, because that's the thing, that's the enzyme that determines how stable the virus is across multiple infections. And so the better this enzyme is, the less likely the virus is to mutate itself out. Use reverse genetics to assemble into a DNA clone. This is the stuff that started already in the year 2000 when we did the first assembly of a cDNA clone of an RNA virus that was a polio virus from scratch. Only a few years later, the Barrick lab did that with a SARS virus. And then you're going to serially pass it in vivo to round out the edges and make everything work just right. Here is a convenient diagram of the possible synthetic route of the laboratory creation of SARS-CoV-2 as drawn by these four authors. Here again, you're going to see the selection. You're going to see the actual steps by which they're doing it. Look here how they're introducing the two cleavage sites, restriction enzyme sites, which allow you to move this receptor binding motif from one place to another. And so now this can be done as you want. That's the cloning vector. Then they're going to put the furin cleavage site in. They're going to engineer the full spike. Then they're going to put that in and make this full-length DNA, and they're doing that, showing you that here. Gibson Assembly is one of these commercially available kits that allows you to take different PCR fragments that you amplify, and Gibson Assembly can assemble those, gives you tools to make the right ends here so that these ends all join up in the order that you want them to. And Gibson Assembly is really a commercially available product that does exactly what they're showing you here. And that's what's cool about this diagram. You don't have to understand all of the things that they're talking about to understand that to a molecular biologist, this is a step-by-step cartoon which shows you how a virus like this could be made from certainly from a template virus plus a laboratory of genomes from which to select these various pieces from. And then again, the introduction of these two restriction enzyme sites to the original template is what allows them to design the receptor binding motif and then put that in and assemble it and then get it into transfections into Vero6 cells, and now you have live viruses that you can then take into animals and passage. So I'm going to read the final remarks of this paper. There's more to the paper than this. The details of the methodologies are a couple more pages that I am not an expert enough to talk about, and also don't think it's really necessary for the purpose of a live stream tonight to draw your attention to this paper. But many questions remain unanswered about the origins of SARS-CoV-2. Prominent virologists have implicated in a Nature Medicine letter that laboratory escape, while not being entirely ruled out, was unlikely, and that no sign of genetic manipulation is present in the SARS-CoV-2 genome. That Nature Medicine paper is the Christian Andersen trash bucket that I have lambasted with a bike ride, and lots of other people have lambasted, including the virologist from Norway who was one of the first dudes with a faculty position to come out and publicly state that he thought that this was a laboratory virus. However, here we show that the genetic evidence within the spike gene of the SARS-CoV-2 genome does exist and suggests that the SARS-CoV-2 genome should be a product of genetic manipulation. Furthermore, the proven concepts, well-established techniques, and knowledge and expertise are all in place for the convenient creation of this novel coronavirus in a short period of time, and that is what I have been saying on the bicycle. The proven concepts, the well-established techniques, and the knowledge and expertise are all in place for the convenient creation of a novel coronavirus of this type. Definitely. Motives aside, the following facts about SARS-CoV-2 are well supported. If it was a laboratory product, the most critical element in its creation, the backbone template virus, is owned by military research laboratories. The genome sequence of SARS-CoV-2 has likely undergone genetic engineering through which the virus has gained the ability to target humans with enhanced virulence and infectivity. The characteristics and pathogenic effects of SARS-CoV-2 are unprecedented. The virus is highly transmissible, onset, hidden, multi-organ targeting, sequelae unclear, lethal, and associated with various symptoms and complications. As you know from yesterday's live stream, this may not be entirely true anymore. We may actually understand the mode of action. The interaction with the ACE2 receptor causes an upregulation of ACE2 and a downregulation of ACE, which sets off a bradykinin storm because ACE is responsible for degrading bradykinin, and bradykinin is a hypotensive actor which causes lots of changes, including capillary permeability, which can lead to a lot of the symptoms that they see, including the stuff in the lungs. So, again, that part might be a little bit hype. Number 4, SARS-CoV-2 caused a worldwide pandemic, taking hundreds of thousands of lives and shutting down the global economy. SARS-CoV-2 didn't shut down the global economy, but we shut down the global economy, presumably because of SARS-CoV-2. It has a destructive power like no other. Again, I think this is also kind of baloney. This part of this starts to bother me because we don't talk about the minors, which I still think are a very important observation that are not discussed here except within respect to the 4991 sequence. But where the 4991 sequence came from is not clear. But I do think that the 4991 sequence probably came from those minors. In that case, one of the things that bothers me about this is covering that possibility up here. Covering up the possibility that this is really where it came from, instead trying to blame the army for it, which is a little bit odd. I think basically it's still not really saying anything about who did it. But judging from the evidence that we and others have gathered, we believe that the finding the origin of SARS-CoV-2 should involve an independent audit of the WIVP4 laboratories and the laboratories of their close collaborators. Such an investigation should have taken place long ago and should not be laid any further. Well, we can already forget about that because any evidence that was there is already destroyed. We also note that in the publication of the chimeric virus, and this was the big chimeric paper that I've covered a couple times, the attribution of the funding of Shang-Chi Li by the NIAID was initially left out. NIAID was funding Shang-Chi Li through none other than Peter Daszak and EcoHealth Alliance. And that was needed to be added as an addendum about a year after, or whenever they put that in. This unusual behavior needs an explanation, and then what is not thoroughly described in this report is the various evidence indicating that several coronaviruses recently published are highly suspicious and likely fraudulent. These fabrications would serve no purpose other than to deceive the scientific community and the general public so that the true identity of SARS-CoV-2 remains hidden. Although exclusion of these details of such evidence does not alter the conclusions of the current report, we do believe that these details would provide additional support for our contention that SARS-CoV-2 is a laboratory-enhanced virus and a product of gain-of-function research. A follow-up report focusing on such additional evidence is now being prepared and will be submitted shortly. So, my friends, this is some big news. This is some big deal. I think this is a really, really cool paper because it's something that we've waited for a long time. It's something that we've been expecting since this girl has been on the news. I've been expecting since Dayu has been lecturing us on Twitter. We've been expecting since Dan's paper came out. It's a really great sort of summary of what's going on here, and I do think there are a few holes. I do think there is a little bit of hype and a little bit of over-exaggeration about the current effects of the virus and where we are now, unprecedented destruction and this kind of stuff. This kind of conjecture doesn't need to be in the paper. But what I do like about it a lot is it kind of ties a lot of the loose ends together. I like the idea that the author from the blog Nerd Has Power is there because I've often felt that this person was underestimated as a person leading the charge here. That Nerd Has blog was Power blog was ignored for a long time, I covered it on a bike ride. I dedicated a bike ride to it, and it was really one of those early publications that really drew my attention as a biologist because it was talking about biology, the non-synonymous mutations, and the striking homology between these sequences was noted by that author. And now what I'm noticing, and I think you should take from this paper, is the very interesting role that the RNA dependent RNA polymerase and the selection of this enzyme would have played in the possible engineering of this virus. And also the reason why Shengji Li and Barrick and these people were always sequencing this particular gene. It never occurred to me until I read this article that this gene, because it is so important in transcribing the genome of the virus during replication, it would also be the enzyme which would be responsible for maintaining the fidelity of the genome, and it would be able to explain why the SARS original breakout in 2003, as Alina Chan showed, went through very many amino acid changes during the initial part of the pandemic or the outbreak, and then slowed down near the end. Whereas this virus is not changing at all. And one reason might be, and this paper posits this, is that the RNA-dependent RNA polymerase is selected specifically because it has such high fidelity and results in such stable viral expression. If this is the case, and we could find this out at some point, if this is the case, then I think this is one of the best smoking guns in this manuscript. The other one is the restriction enzyme sites, the restriction sites that border the receptor binding motif, which from a molecular biology perspective facilitate the removal of and insertion of new pieces of DNA at those restriction sites. And this really is something very special if this is really true. So I highly recommend that you read the paper. The paper is titled Unusual Features of the SARS-CoV-2 Genome Suggesting Sophisticated Laboratory Modification Rather Than Natural Evolution. And they're going to give you the delineation of its probable synthetic route. That means they explain to you how they think it could have been generated in a laboratory. So they take two birds with one stone. It's a very well thought out paper. It's a very well cited paper. They cite everyone whom I've worked with and interacted with on Twitter. Rosanna and Yuri's paper is cited there. Mona's preprint is cited there. Dan's paper is cited there. Alina Chan's data is cited there. Petrovsky's sequencing data is cited there. All of the papers that I have covered on my bike rides in the past 6 months are basically represented in this paper. It's a very spectacular print. So if I can recommend it highly enough, please read the paper. I'm going to cut this video and put it up for tomorrow. Thank you for joining me. If you can, please support this stream with the links below. I just lost a GoPro last week on the street. It's one of the reasons why the bike rides have not come out recently. The bike rides will come out again, but just to let you know, GoPros are expensive. I did not know when I lost it. It was my back camera GoPro. And so I went back looking for where I thought it would be, but it definitely wasn't there because, you know, when you find a GoPro on the street, you definitely don't give it back. So those bike ride videos are coming back very, very soon. I've got a couple in the pipeline right now that just need final editing. Again, if you liked it, like and subscribe. Please share this video and this channel with your friends. This is Gigaohm Biological. This is high resistance, low noise information stream. It's a biological information stream brought to you by a biologist because you know what? Biology is the truth of our lives. We are animals. We live in a biological world living by biological rules, and it's fine and high and mighty time that we all learn those rules, because the best part about it is if we all understand biology, we can all think for ourselves. We don't need experts. And I'm telling you right now, just like you can learn how a car works, just like you can learn the physics of a compound bow, you can definitely learn basic epidemiology. You can definitely learn basic immunology, and you can definitely learn all about coronaviruses. This coronavirus isn't that special, although they tried to make it special. As you can see from this discussion, it is very likely that they have selected an RNA-dependent RNA polymerase that is very effective and highly not error prone. It also may be possible that they selected a virus from 2012 that was especially attractive to them for any number of reasons, including the fact that its receptor binding motif had nearly complete restriction sites available so that they could easily swap this motif from place to place, and so it made it a very convenient backbone. And that's another thing that this paper really puts out. So I think these points are really, really valid. I think they're really interesting ways to tie together all of the data and all of the observations that various authors and various investigators have made over the last 6 months, ties them together very neatly into a succinct set of ideas. I'll probably revisit this paper again and again because it is that good, and as we move forward, we're going to get more data that will either confirm or deny this. And don't forget, besides this paper, the other paper you really need to read is the Bradykinin hypothesis paper, which was the one that was done by the Oak Ridge Laboratory in Tennessee, which I covered yesterday on my live stream, because that does explain what this paper still calls an unexplained, multi-systemic manifestation of a disease. Actually, it can be explained quite easily by this supercomputer simulation and the results that came from it. So I think what we're going to do over the next few days is probably just talk about current events, talk about the things that are happening around the world, and then at the same time in the second half of each of my streams, I'll just try to bring this paper together with that computer modeling paper as we continue to update, which I think is becoming a very clear picture of what's happening in the world. The pandemic's over, folks. The only thing that's left now is the psychological damage, the psychological hole that we're in. We're going to pull each other out together by looking for the truth together, by reading, by critically thinking, and by changing the minds of everyone around us one day at a time. I love you guys. Thank you so much for joining me. Thank you for sharing this video with everybody that you know, and hopefully I'll see you very soon again. Thank you, thank you, thank you.